Hi, right now I'm going to explain some things about vaccines. This is meant to hopefully improve your knowledge on vaccines in general, to help you understand that they are safe and makes life safer. Then hopefully you can pass this knowledge on to someone else. This is especially important now considering there are many people out there who are refusing or hesitant to take the COVID vaccine because they are skeptical of how it was made. A vaccine is a substance used to stimulate the production of antibodies and provide immunity against one or several diseases. It is first administered. Vaccines contain weakened or inactive parts of a particular pathogen that will trigger complementary lymphocytes to rapidly divide to create a large number of the specific lymphocytes. Lymphocytes will then turn into plasma cells and memory cells. The plasma cells will secrete antibodies to combat the pathogen, and the memory cells will retain information relating to the pathogen and antibodies formed. If a person were to encounter the authentic pathogen, it will result in the memory cells to rapidly divide to produce large amounts of antibodies to remove the pathogen before severe symptoms develop. Here are some extra definitions of terms used. I am now going to compare the development of the Pfizer vaccine against the general development procedure for previous vaccines. Begins with the exploratory stage, which involves basic laboratory research to discover a possible antigen, which is a stimulant that is capable of triggering an immune response in the body to produce large amounts of antibodies to destroy any dangerous pathogen. This takes between 2 to 4 years. For the COVID vaccine, researchers have been studying coronaviruses for over 50 years. This meant scientists have existing data on the structure, genome, and life cycle of this type of virus. This current virus we are experiencing is a member of the coronavirus family. The preclinical stage includes experiments with cells, tissues, and animals over 1 to 2 years to determine the safety of the trial vaccine, its effectiveness in triggering a desired immune response, effective dosage amount, and method of administration. For the COVID vaccine, early efforts by scientists at Oxford University to create an adenovirus-based vaccine against MERS provided the necessary experimental experience and groundwork to develop an adenovirus vaccine for COVID-19. The first phase of clinical trials takes around 2 years and usually entails the vaccine being administered to about 20 to 80 volunteers. The aim is to assess the vaccine's efficacy, side effects, sufficient dosing, and immune response. Phase 2 involves a larger group of several hundred individual participants. Some of the individuals may belong to groups at risk of acquiring the disease. These trials are randomized and well-controlled and include a placebo group. The aim is to begin tracking the efficacy of the vaccine, the doses of the vaccine, the duration of immunization, and the delivery mode of the vaccine. Phase 3 includes testing over a much larger group over the course of 5 years to test efficacy and safety due to the possibility of detecting a rare side effect. In the clinical trials of the COVID Pfizer vaccine, the 3 phases were performed simultaneously, as well as the high interest in volunteering to participate in the clinical trials made the process faster, and this is not an unrare occurrence. 43,000 participants enrolled in the Pfizer clinical trials, with consistent efficacy across age, gender, race, and ethnicity demographics. There was observed efficacy in adults over 65 years of age was over 94%. According to the US FDA, 3,000 participants are already sufficient for standard phase 3 trials. Their similarity is that all past vaccines and the current Pfizer vaccine meets the high standards for safety, effectiveness, and manufacturing quality the FDA requires of an approved product. Now I'm going to give a brief timeline of the history of vaccines. In 1796, Edward Jenner inoculated a 13-year-old boy with cowpox and demonstrated immunity to smallpox. In 1798, the first smallpox vaccine was developed. Over the 18th and 19th centuries, systematic implementation of mass smallpox immunization culminated in its global eradication in 1979. Louis Pasteur's experiments spearheaded the development of live attenuated cholera vaccine and inactivated anthrax vaccine in humans in 1897 and 1904. The plague vaccine was also invented during that time. In 1923, Alexander Glennie perfected a method to inactivate tetanus toxin with formaldehyde. The same method was used to develop a vaccine against diphtheria in 1926. Pertussis vaccine, its development took considerably longer, with the whole cell vaccine first licensed for use in the US in 1948. Viral tissue culture methods was developed in 1950 to 1958 and led to the advent of two different polio vaccines. Now I'm going to explain the impact vaccines have had throughout history, and this image provides evidence. Firstly, it has completely eradicated smallpox globally, which was the cause of 500 million deaths. It has also reduced measles cases by 73%, preventing 23 million deaths. Now, that's with vaccines. In the United Kingdom and Northern Ireland, when there was a drop in pertussis vaccination in 1974, what followed was an epidemic of more than 100,000 cases and 36 deaths. Reduction of the same vaccine transformed 393 cases and no deaths in 1974 to 13,041 deaths in 1979 Japan. Thank you.